is that the conversation's going on down here. His graphics make sense if you have the kind of prescription drug coverage he has. Not sure what that's supposed to represent. Neither is anybody else. For a long time, there was investigation of natural treatments. People would go off into the jungles of Asia and South America and Africa and talk to the locals and try to figure out what they were doing to treat various ailments. And then they would get samples and bring them back and test them to see if they worked. And they generally don't. And when they do, they don't tend to work particularly well. But when they do, then it's a question of, so what's happening here? Is what's in here that's causing an effect? So those are the natural sources. Natural sources have shifted. Now natural sources, instead of going off into the hinterlands of the jungles and talking to the indigenous people, is that people who work in drug research read a lot of the basic, what's called pure science research, where somebody has taken a look at a particular type of, say, kidney cell, and figured out a metabolic process that happens in the kidney cell. And somebody at the drug company will go, that's probably connected to this issue having to do with kidneys. There's no treatment for that issue, or there's no good treatment for that issue. Maybe we have a target for a drug molecule to get in there and fix things. In some cases, what you're looking at are what are called bioactive compounds. Molecules that have some sort of activity in a biological system, and now you've got to come up with a compound that will somehow adjust that, possibly block it, possibly enhance it. And these are big, complex molecules. It's not that easy to go after. And sometimes it isn't enough to go after a single process. There's, as we've talked about before, it is difficult to treat for viruses because viruses typically do processes that your cells are doing all over the place, and blocking those would be bad for the other cells that don't have viruses in them. But there are, excuse me, processes that are specific to viruses, but even then, the only really effective antiviral treatments that are on the market right now are go after a couple of targets simultaneously. They're going after single targets in something as fast reproducing as a virus is, it tends to evade. You go after two things at once, it's tougher to do. Three things at once, which is what they do with HIV, it's tougher to evade. Once you've got a molecule in mind, you have to think about, okay, this is a molecule that could have an effect. Where's it going to have the effect? Well, it's going to have an effect in these cells. How do we get it into those cells? There is no way in the world that we can get this molecule delivered to those cells in any kind of effective dosage. So there is delivery issues, and there's what they call bioavailability. Can we get it into a person at levels that are actually usable? Or does, oral medications have to get through the digestive system, have to get through liver processing. Injectables have to deal sometimes with immune responses if you've got a fairly large molecule. They need to be able to get through the cell membrane. They may need to be able to get through the nuclear envelope. They may need to get into the part of the cell where whatever's happening is happening. These are all things that have to be dealt with in terms of theory. And then at some point, or you look at molecules, and there is some use of computers to look at the tertiary structure or quaternary structure of large molecules and play with it this way. Maybe we can get this to happen, maybe we can get that to happen. But basically, eventually what you're moving toward somebody at the front end comes up with a potential molecule that could be useful in a pharmaceutical. And they go through the literature, they talk to everybody else that's dealt with these kinds of things. Is there already a patent out there on something similar enough that it's going to be an issue? If there's not, we need to get to the lawyers and get a patent put in on this. Because we don't want to be starting or in the middle of research on our molecule and not have the right to the molecule if it pans out. So you put a patent in place. Once granted, the patent lasts 16 years. The research may take 12 before it's a marketable material, if it ever is. 9 out of 10 patents are on things that never ever make it to market. For one reason or another, at some point in the system, they just crap out. They wind up not doing what they're supposed to do, or you can't come up with a deliverable system, all the different things. And also, you have to think about, can we make this stuff? Can we make this stuff at levels where it is something that we can make our money back on and make enough of it that for the number of people that have this issue, we can deliver it to them. So there's a whole lot of things that have got to be done. Drug patents are not renewable. Most other patents are renewable. They decided in the 1970s, I think, that by cutting the patents off at 16 years after the patents lapse, generic companies with a certain amount of regulatory clearance could make their copy of your patented molecule and sell, what are called generic drugs. Generic drugs don't have to be as effective as the original. They have to hit a certain threshold, somewhere in the upper 70s, I think. I read about this and I've forgotten the number. And why wouldn't it be equally effective? Because there's a lot of other stuff in a dosage. There's a whole bunches of molecules in pills to help them get through your digestive system and get them into your blood and get them into your cells. And those don't have to be reproduced by a generic. They just have to be available, they have to have a certain level of effectiveness. A drug company has many options of combinations to get a pill to work as perfectly as they can get it to work. The generic companies don't invest in that many options. So they fiddle through their limited catalog to find something that will hit the FDA's threshold for generics, which is why generics, some people go, you put me on the generic, but it's not working the same way, or you're putting it on me on the generic and it's doing this weird stuff to me. Well, there may be something in the generic pill that's not in the actual pill, other than the drug itself, that could be either tamping down its effectiveness, 78% effectiveness means 22% ineffectiveness. Or there could be a lot of other things going on. And then you run into issues across national borders and who's respecting your patents and all this other stuff. Right now, there's a tremendous amount of research into what are called biologics. These are large-scale molecules that are adapted from large-scale molecules that work in cells. And it's a very trendy area, but also one of the reasons for biologics is that biologics turn out to be difficult to manufacture. You have to come up with a whole manufacturing system for them. And once you've patented the drug, you don't necessarily have to patent the manufacturing approach. until you've gotten it all the way through and found that it actually works. And now you've got a patent on production that's going to last another 16 years. Or the process is too expensive for a generic company to invest in. They're not going to make enough money, setting up. And so some of these very, very expensive drugs that never seem to go off patent, they never seem to go generic, that is part of the reason for that. Testing. Supposed to be a T in there. You do what's called in vitro, Latin. These are tests outside of of a living system. Yeah, cell culture is considered to be in vitro, and cell cultures can be very useful if you have the right kind of cells, which is not that easy to find. You can't culture every single type of cell. Also, a cell culture is not, like if you have a cell culture of spleen cells, they don't behave completely the same way that they would in a spleen. Is the interactions with the surrounding architecture and the body as a whole are different. But you can get some idea if you think this is supposed to work in a cell a particular way. A cell culture may tell you whether you're right or wrong about that. Is it So it's a good first place. After the cell cultures, you generally move into animal models. For that, you need animal models that will mimic humans. So if you're going after a particular condition, you need an animal that will have some reliable version of that particular condition. So in some cases they deal, I think mice don't get a certain thing, you have to deal with something that does. With COVID, it turns out mice are not particularly supportive of COVID, but hamsters are. And so there was a certain amount of research done on hamsters. Sometimes there is no good animal model, in which case it's tricky to get through. Once you get into this level of tests, there's a point at which you're going to have to get clearance to go to the next step. You have to send your data to the FDA, they look it over and go, okay, this doesn't look to be that toxic, it looks like it's working, we will allow you to get into clinical trials. Also, there's been a certain amount of computer simulation work. Remember, computer models, cell cultures, and animals. Computer simulations tend to be way up front. And of course, they're limited by our limited knowledge. There are mice that are developed that show conditions they don't actually get. There's like mice that have a mimic of multiple sclerosis. Mice don't naturally get multiple sclerosis. And so that's the issue, is that really an accurate version? What will work on them? Will it work on multiple sclerosis in people or in any other animal exactly? There's also what they call knockout mice, where you can shut down a system. In mice, you can shut down their immune systems so that you can do transplants of certain things. There's, again, a lot of limitations. So the FDA goes, okay, we like what you're doing so far. You can start human experimentation. Those are clinical trials. When you hear somebody go, this has been clinically tested, maybe that's true. Doesn't mean that anything came of that. Doesn't mean it works. Just means that it was tested. And sometimes they go, it's been shown in clinical trials to do certain things. A lot of times that's a lie. Is that you can claim that you've done stuff in clinical trials and lie. It's false advertising. If somebody complains to the Federal Trade Commission, maybe you'll get slapped on the wrist. So a lot of times with these nutritional things claim clinical testing, sometimes they're phrasing it so it's not completely a lie, and sometimes they're just lying, figuring it's not. Nobody's going to complain about it. Nobody's going to check. But clinical trials are technically trials on people that have been reviewed by the FDA, and there are 3-4 levels of clinical trials. These trials need to generally they have to be done out in the world. Generally, you're not gathering people into your pharmaceutical lab somewhere. You're going off and so you're contracting with hospitals and doctors to do this. You need informed consent. You may or may not need control groups. I'll talk about that as I go through the trials. But there's a lot of stuff that you're going to need. I'm just looking. I think before I get into the actual clinical trials, phase 1, phase 2, phase 3, and phase 4, I'm going to go to another video.